welcome everybody. My name is Professor Peter Nash. I work at the School of Medicine at the Griffith University in beautiful downtown Brisbane, Queensland, Australia. And I welcome you to the symposium. We're going to be looking at JAK inhibitors in particular. We're going to be looking at selectivity, and we have we're very fortunate to have one of the world's authorities, not only rheumatoid arthritis, not only in developing a vaccine that'll put me out of work, but John is a wizard as far as JAK inhibition is concerned, and will fill us in on all those details about selectivity. And he comes from we're very fortunate to have him. He comes from Newcastle University in the UK, where he directs the therapeutics. He directs research for the entire hospital, not just rheumatology. He works on the Newcastle Biomedical Health Innovative Institute, and we're so glad to have you with us. So welcome, John. Nice to see you, and we hope COVID has treated you well. Here are our disclosures. We've been doing clinical trials for, I think, 30 years now, including all biologic agents and targeted synthetic agents since they were invented. And John similarly has been working very hard educating industry. So they're the disclosures. And next slide, if it will work. We'll pass over to John to show us his presentation, please. Welcome, John.
Thank you. Thank you, Peter. And I'm very grateful to be with you this morning. Yes, I'm from Newcastle upon Tyne in the northeast of England. For those of you who don't know it, lovely city. Not quite as sunny as where Peter's sitting right now, but lovely nonetheless. So yes, so the title of my talk is, JAK inhibitors, are they the same or are they different? And you will be getting experience in the clinic, and you'll be asking yourselves whether these drugs are the same or whether they're different. And what I'm going to do is focus very much on the mechanism of action and the selectivity of these drugs, and indeed addressing this question. So I've divided the talk into 4 sections. First of all, just some basics about JAK inhibition and how these drugs work, which I hope will be revision for most of you. The main chunk of it sounds maybe not fascinating, but it's pretty important, which is, in the lab, can we see differences between these drugs? And I'll be surmising on the clinical relevance of this as we go through. I'm then going to talk about a relatively new drug, which really is different to the others because of the way that it works. So I thought I would talk about it in this presentation. And then a couple of slides on COVID at the end, again, because it does bring out differences between the different drugs. So let's get started with some homework, really, and some revision. So protein tyrosine kinases, that's what JAKs are. And tyrosine kinases are enzymes that transfer a phosphate group from ATP onto a protein. So here's a protein, here's ATP. There's a protein kinase here which catalyzes the reaction. A phosphate group tags onto that protein, and this usually switches function on or off, and is often part of an enzyme cascade. Typically, they associate with membrane receptors and provide key steps in signal transduction, but because it's such a useful mechanism, there are a lot of them. There's 518 tyrosine kinases in the human genome, and they're tractable drug targets. And yeah, that's all I'll say right now on that. So here's the JAKs. There's 4 of them, and so they are a family of these protein tyrosine kinases. We call them JAK1, 2, and 3, and TYK2. They're structurally very similar, as you can see, but they have subtle differences in the structure of their ATP binding pocket. So in order for them to take this phosphate from an ATP, the ATP sits in a little pocket within the enzyme or within the enzyme, and they're slightly different, which means that they're targetable with drugs, theoretically selectively. So you should be able to develop a drug which maybe targets the JAK1 mechanism more than the others. So this is how they work, and again, I suspect this is revision. They tend to be involved in cytokine or growth receptor signaling. So here's your cytokine receptor, here's your cytokine, and here are the JAKs that associate with the receptor. When the cytokine binds its receptor, the JAKs phosphorylate one another. After that, the JAKs then phosphorylate the tail of the receptor, which becomes a binding site for these STAT proteins, which is why we call it the JAK-STAT pathway. And the JAKs also phosphorylate the STATs. And once the STATs have become phosphorylated, they head off into the nucleus where they become transcription factors. And they're largely involved in the inflammatory response or growth factor signaling. And as I've already implied, all of these steps require tyrosine phosphorylation. So a whole series of tyrosine phosphorylation steps, which is why these enzymes, these JAKs, are so critical in immune responses. So this is the human kinome, and I've mentioned these 518 different kinases. And up here at the top, this is the JAK family, but you can see there's many of these. And so this is a bit of a challenge in drug development because they all have these ATP binding pockets largely. And so to develop drugs which just target one family rather than another is obviously challenging, let alone selectivity within a family. Just as some examples, I found this. So here are various tyrosine kinases and the drugs that target them. And what you're seeing here is the drugs mostly sitting in ATP binding pockets. And for example, here's tofacitinib, and this is what the JAK1 binding pocket looks like, and this is a JAK3 binding pocket. So you can see that these drugs are very cleverly designed to just sit in the binding pockets of the enzymes. And here's a bit easier to see. This is tofacitinib binding, I think this is JAK1, it could be JAK3, I can't remember honestly, but what you can see here in green and red, that's where ATP usually sits to pass on that phosphate group, and in pink and blue is tofacitinib. So basically they wouldn't both be there together. Tofacitinib would sit there and displace the ATP, so the enzyme basically cannot work because there's no phosphate to transfer. So that's how these drugs work, and I'll come back to this theme. In terms of the various pathways, again, I hope that you're familiar with this sort of figure. I've illustrated 6 different families, if you like, of cytokines here. And what you'll see is mostly the receptor comprises heterodimers of 2 different JAKs. So for example, what we call the gamma chain cytokines, which are very important in T cell proliferation and survival, IL-2, IL-7, IL-15, for example, they depend on JAK1 and JAK3, the type 1 interferons, JAK1 and TYK2, IL-6, JAK1 and JAK2 with a bit of TYK2, interferon gamma, JAK1 and JAK2, IL-12, 23, JAK2 and TYK2, and the only homodimers of JAK2 are the hematopoietic growth factors, so erythropoietin, thrombopoietin, GM-CSF, etc. As I'll come on to, what's interesting, we don't understand very well the balance between the 2 JAKs. In other words, does IL-6 have exactly the same dependence on JAK1 as IL-27, or does one of them maybe signal more through JAK2? And I'll come back to that in a second. Now, going back to the selectivity, it is possible the clever medicinal chemists to synthesize or create small molecule drugs which can to a certain extent be selective. So Paracetamy, which I'm sure you're all familiar with, is more selective for JAK1 and JAK2. Having said that, because all of these pathways involve JAK1 and JAK2, it makes it a relatively non-selective drug for that reason, because all of these pathways involve either JAK1 or JAK2. So tofacitinib, for example, is JAK3 more than JAK1, but also JAK2 and a little bit of TYK2. So again, it's pretty non-selective. There are then some JAK1 selective drugs, such as filgotinib and upadacitinib, which will theoretically at least not block these 2 right-hand pathways. As I'll come on to towards the end, there's TYK2 selective drugs, again, with a different pathway selection. And in Japan, there's a drug called peffercitinib, which is JAK3 selective, and so only blocks this pathway. So what does this all mean? What's the rationale? Well, The rationale depends on knowing which JAK pathway you want to block for benefit and which one you want to try and avoid because perhaps it's responsible for side effects. So, for example, if you think JAK1 is the pathway you want to block, and we will come back to this, and perhaps JAK2 is the one you want to avoid, if you can create a drug which is selective for JAK1 and less selective for JAK2, it should be possible to find a dose of the drug which largely provides benefit without toxicity. But the really important message from this slide actually is, of course, as the dose goes up and as the concentration within the cell goes up, you will start to block JAK2 as well. And I say that because we've become very used to monoclonal antibodies over the last 10, 20 years. And monoclonals are specific. They only bind one target and they can't bind anything else because of the way they're designed. Because these are small molecule drugs, specificity doesn't really exist. We only can talk of selectivity because they will start to have off-target effects. And indeed, with those 518 kinases in the kinome, you may even start to see off-target effects in other families at very, very high doses. So that's the concept of selectivity. I'm going to show you this very briefly, and I'm going to come back to it. It looks a bit tricky, but all we're showing here, so this is a very specific, we're looking here at IL-21. And we're stimulating NK cells with IL-21. And as a readout, we're looking at phospho-STAT3 because this is one of the stats that gets phosphorylated by IL-21 via JAK1 and JAK3 inhibition. And what these horizontal lines show is the amount of baricitinib or upadacitinib or tofacitinib to inhibit by 50% the phosphorylation of STAT3 by IL-21. So that's what we call the IC50. It's a pharmacological term. It's a convenient term for understanding and comparing drugs. And then these lines here are the 24-hour profile of the drug in question at the doses shown here. So baricitinib, we usually use 4 milligrams daily. Upadacitinib, we usually use 15 milligrams daily. Tofa, it's usually 5 milligrams BD. And what you can start to look at is how much of the day the drug is above the IC50 concentration. So for example, in this particular example, IL-21 in NK cells, tofacitinib, even at 5 milligrams BD, is above 50% inhibition for 61% of the day, or 17.5 hours, whereas over here, baricitinib scarcely gets above IC50. So even at 4 milligrams, just one hour and 20% on average inhibition. So you see, this is where the selectivity starts to come in. You're just looking at a particular cytokine in a particular cell, but you can start to compare the drugs in terms of their blocking capacity. And I will come back to this later on. Having said all that, I found this paper, which is comparing baricitinib and tofacitinib, which do have a different selectivity profile. So Bari is JAK1 and JAK2, tofacitinib is JAK3 and JAK1 with a bit of 2. And here in this paper, they look at all sorts of different in vitro immunology. So the maturation of dendritic cells with LPS, and you can see this is with baricitinib 300 nanomolar. Here's tofacitinib 300 nanomolar, and the expression of CD80 and CD86, which are maturation markers. Actually, they both block to about the same degree. PDC production of interferon alpha, which is important immunological profile, blocked about the same. B cell differentiation measured in various ways, blocked about the same. T cell proliferation and differentiation. The reason I'm showing you this slide is, despite theoretical differences between the drugs in those, very specific assays that I've shown you, when you start to look at whole cells, the differences may look less. I'm now going to focus on a paper that came out last year, and this was a publication from Galapagos, which is the company that originally developed filgotinib. And again, I want to show you this paper. It's a bit complicated. It was a big paper. It took a lot of reading. If you include all the supplements, it's 45 pages and a lot of data, but it's interesting and it made me think, and I'm going to try and make you think about what this all means. It's a little bit similar to the slide I showed you a couple of slides ago with these 24-hour profiles. But in this paper, they take blood from RA patients, they look at mononuclear cells, they look at whole blood. And if you remember, what I showed you was IL-21 in NK cells, looking at phosphostat-3, I think it was. In this paper, they look across all immune cells using all the different drugs with different cytokine stimulations, and they try and compare the four drugs that we're all familiar with in terms of their ability to inhibit different pathways in different cells in response to different cytokines. And it is complicated. This is just one figure from that very long paper. And there are lots of figures that look like this, again, looking at different cells, different cytokines. But I'm just going to take you through this a little bit. If you look here at the top right, now this is interferon alpha stimulation. And the drugs here are filgotinib, baricitinib, tofacitinib, upadacitinib, and it's in CD4 T cells, this example. And actually here, the drugs look pretty similar in terms of their profiles, perhaps a little bit less here for filgotinib, but overall, over 24 hours at the doses we use, the inhibition is similar. And I'm going to come back to this. And on this side, IL-6 phosphostat-1 in monocytes, again, not that different. They look fairly similar. But as you maybe come down here to JAK1, JAK2, so this is interferon gamma in neutrophils. Here, for example, filgotinib scarcely it doesn't reach the IC50, so you've got values of 0. And you can see, for example, upadacitinib looks like it's going to be blocking this pathway more than the other drugs. So you can imagine that with many, many, many pages of figures like this, you can start to get a profile of the different drugs. Here's JAK1, JAK3, which is that T cell differentiation, maturation pathway. And again, you can see upadacitinib and tofacitinib quite well above the line, whereas baricitinib and filgotinib just about touching the line. So, and other examples, JAK2, JAK2. So this is the GM-CSF. UPA seems to block it, but not the other JAK inhibitors. So I'm sorry to go through that in detail, but I think it's quite important. And as I say in this paper, they look at all sorts of different cytokines and all sorts of different cells to try and get a feel for the different drugs. And some interesting data come out of this. So for a start, it depends on whether you're looking at the whole blood or PBMCs. And the reason is in whole blood, these drugs bind to proteins, to serum albumin to different extents. And that changes the ranking in terms of their selectivity. Surprisingly, depending on what cell type you look at, you get slightly different results. So the same JAK inhibitor does not block the same pathways identically, even in response to the same cytokine in different cells. In the same cell, you might get a different response depending on whether you're stimulating with one cytokine or another. So maybe IL-7 versus IL-15 may give you slightly different responses in T cells with the same JAK inhibitor. And whether you read out through STAT1 phosphorylation or STAT3 phosphorylation may look a bit different. So the overall message of this paper, well, I'll come back to the overall message. The overall message for me is that it's not quite as straightforward as you might expect it to be. So the more complicated your system, the less predictable the inhibition. Having said that, you can develop a general hierarchy, and that's here on the right. And up the top here, I showed you in that previous diagram that all the JAK inhibitors looked very similar when you stimulated cells with CD4 T cells within, here it's monocytes actually with interferon alpha, and measured JAK1, JAK2 inhibition. So this is why they've all got 1. And then the darker you get, the more selective. So for example, down here, filgotinib scarcely blocks the JAK2 pathway, the number in that box, I think, is 16.6. So compared with its inhibition of interferon alpha here, it's 17 times more selective for interferon alpha, and it doesn't block this GM-CSF pathway very well at all, compared, for example, with Barry, which is 5 times more selective, and UPA, which is 6 times more selective. And you can see across these different pathways, different selectivities. So for JAK1, JAK3, I showed you the interferon alpha and IL-6, there wasn't much difference between the drugs, and that's why even for IL-6, you've got 1 to 2-fold differences. As you get to JAK1, JAK3, you can see here that UPA's 5 versus 1.8 for TOFA. You start to see differences emerging, and the further down, the more differences there are between the drugs. Again, just seconds on this slide, it shows you all these different pathways in all the different cells in response to different cytokines. And what can I bring out here? NK cells, interferon alpha, STAT5, 69% inhibition. NK cells, interferon alpha, measure STAT3, only 35% inhibition. So it's the same cytokine in the same cell, different readout. You can see quite different results. But the overall message here is the red is inhibition. So whereas blue is not inhibition, and you can see differences between the drugs. And I think the other thing to recognize is, as what I've said, is as the dose goes up, here's TOFA at 5 milligrams and 10 milligrams, obviously you're getting more red, and you're seeing red in areas that were previously blue, which is this slight loss of selectivity as the dose goes up. And I think that's quite an important thing to notice, that obviously we don't use 10 milligrams BD of tofacitinib, but that's partly why, because the selectivity is lost, which means you could start to see effects that you don't want. And this is a similar map, just showing time above the IC50 at the clinical doses. So I'm sorry, a lot of sort of lab science there, but I think there are some important messages, and those are on this slide. So first of all, does blood reflect tissue? In other words, all that 45 pages of work was done on peripheral blood from rheumatoid arthritis patients, from healthy controls. But actually, what we really want to know about is what's happening in the joints, and whether these drugs work on rheumatoid will depend on the concentrations in the synovium. And of course, that depends on the pharmacokinetics, how much you absorb, how you metabolize, how you excrete the drug, where it's distributed to. And this varies between patients. Pharmacogenetics is very important. And of course, adherence is very important. So again, these aren't monoclonal antibodies where once they're in the patient, they do what they do. They're pretty complicated drugs. The thing that really struck me about this work, though, and probably I've dwelt on it a little bit too long, but why IC50? IC50 is a pharmacological term. It's useful in drug development because it does allow pharmacologists to develop drugs, compare drugs, and indeed look at selectivity. But what those heat maps showed you is that these drugs are blocking numerous pathways in numerous cells in response to numerous cytokines. Is IC50 really important? What if only 10% inhibition was necessary to have an effect? And as I was developing this talk, I was thinking about genetics, and we have genetic knockouts of, for example, JAK-STAT pathways, and they're lethal. So we know we can't knock these pathways out completely. And when you think of rheumatoid and you think of the genetics of rheumatoid, you have 100 different SNPs which contribute to rheumatoid. On their own, none of them do very much. But actually, when you put them all together, they cause a disease called rheumatoid arthritis. So lots of subtle effects can add up to big effects. And finally, the thing I'll come on to is which pathways do we want to block anyway? But just thinking about this IC50, again, I apologize, back to this figure, but look down at the bottom left here. What if 20% inhibition was all that was necessary, or indeed 10% inhibition? You'd go from a situation of selectivity where, for example, filgotinib was not having the same effects of the other drugs, to a situation where actually they were all doing pretty much the same thing, because if you block by 10%, they're all blocking by 10% for much of the day. And that would go for many of these pathways. And my point is, 10% inhibition may only block the pathway for 2 or 3 hours a day, but if you're blocking 100 different pathways for 2 or 3 hours a day, that's likely to add up to something pretty significant. So I'll just leave that thought with you in terms of what these drugs need to do to have a pharmacological or pharmacodynamic effect. And the other question is, which cytokines do we want to block? You know, this is a nice slide from Yoshi Tanaka's recent review in Nature Reviews Rheumatology, showing signaling which is susceptible to JAK inhibition. And there's, you know, we're not showing inflammation or pain on these slides. It's literally just showing all the cytokines which JAK inhibitors block. I think you'd probably agree IL-6 is pretty important in terms of cytokines which are important in rheumatoid, but many of these are. And many of them are dependent on different JAK-STAT pathways. So I think there's an important question there. Which pathways do we block? Some might say that GM-CSF is a bad pathway to block because it could lead to neutropenia, but there's actually anti-GM-CSF antibodies being trialed in rheumatoid arthritis, which are pretty impressive effects. And so I don't think it's immediately obvious anyway which pathways we would like to block. And of course, as I've already mentioned, there's then the individual patient variability, which will lead to differences in concentration, affecting all, you know, as we get older, we excrete drugs less. There's polymorphisms in the way drugs get into cells, polymorphisms in metabolism. So again, as doctors, we have to, of course, look at the individual patient and look at how they're behaving. Just to finish off, I will now touch on some different drugs which do act differently. So the drugs I've mentioned so far, the JAKs that you're familiar with, as I've already mentioned, they sit in the ATP binding pocket, and that's how they work. Dukravasit is a different JAK inhibitor. So this is a TYK2 inhibitor, and this is TYK2 up here on the right. And it's a bit different because it's got this pseudokinase domain, and that's where dukravasit binds. And the pseudokinase domain regulates activation of the catalytic domain, which it sits next to. And dukravasit stabilizes this pseudokinase domain so that it allosterically inhibits activation. So basically, it's a JAK inhibitor, but not by sitting in the ATP binding pocket. It's got an allosteric mechanism of action. And it makes it very specific because this selective, I should say, it's highly selective in this case because of its mode of action, because the other JAKs don't have this pseudokinase domain. So it's highly selective and potent for JAK2. And it's being looked at in psoriasis and psoriatic arthritis. And on this next slide, again, if you remember where TYK2 sits, it's particularly the IL-12/23 pathway and type 1 interferons, both of which we know are important in psoriasis and psoriatic arthritis, and possibly a bit in this IL-6 pathway also. And just one data slide really. This is a trial that Phil Mees published just recently in the Annals in psoriatic arthritis. And this is looking at ACR20, and this is looking at HAC. And as you can see, both doses of dukravasit in this trial, I think it's a phase 2 trial, gave ACR20 responses at 16 weeks of about 60% and about a 0.4 to 0.5 reduction in HAC. The other thing I've just put in for fun is baricitinib as an antiviral. And this goes back to my point at the beginning of the kinome, you know, that you get what we might call an off-target effect because these drugs have the capability at certain doses of inhibiting other kinases. And here we're looking at strangely called NUM-associated kinases. And these activate a scaffolding protein, which is important for viral entry. So these NUM-associated kinases down here are important for viral entry. And as it happens, baricitinib blocks these NUM-associated kinases. You can't see this, it's too small, I can hardly see it on my laptop. But actually baricitinib is between 100 and 1,000 times more potent at blocking these pathways than the other JAK inhibitors. And the data down here simply show in an in vitro model of liver cells, baricitinib does indeed block viral replication. And as you know, baricitinib's been trialed in COVID. This was the ACCT2 study of baricitinib and remdesivir, 1,033 patients. everybody got remdesivir, but was then randomized to either baricitinib or placebo. And the primary outcome was hospital discharge. And in fact, what you can see here, this is days and this is proportion recovered. There was a significant effect of baricitinib, which is quite subtle, p-value of 0.03. But actually in this score 6, which you may remember is high flow nasal oxygen or non-invasive ventilation, there was actually quite a big difference between baricitinib and placebo in hospital discharge. There wasn't any change in risk of death, at least not significant, but certainly delayed discharge. So the primary outcome was the time to recovery. So that's my last slide. So to conclude, the JAKs have different selectivities, at least in vitro. And so you can go from simple enzyme assays, incredibly pure assays, to the whole human being, which is obviously significantly more complex. This certainly translates into distinct ex vivo and in vitro profiles, therefore, but the challenge for me is to decide which, if any, aspects matter in the clinic. Pharmacokinetics and pharmacogenetics both influence tissue concentrations and therefore ultimate JAK inhibition profile. We're in an age of precision medicine, so there's something in the back of my mind here, which is, will we ever reach a precision medicine agenda with these drugs? In other words, between the drugs, are we likely to see different drugs for different patients? I've mentioned dukravasit briefly because it does have a distinct mechanism of action to the other JAK inhibitors, and baricitinib for fun I've included here because of its, if you like, off-target effects on NUM-associated kinases, which associate with antiviral activity. And with that, I'd like to thank you for your attention, and I hope I haven't blinded you too much with the laboratory data. Thank you.
So thank you, John. That was superb, setting the scene. Please ask your questions in the Q&A section rather than the chat section. We've got a question which fits perfectly with the next talk. It's from Edgar Stemler, and it's more a comment really. Regardless of selectivity in cellular assays, provided the scientists in the lab did an excellent job and developed a good binding agent for the active site in the selected JAK work as a matrix, the question arises when administering the drug in vivo, whether the whole network of the immune system is influenced by the JAK inhibition, and therefore clinical data at the end do count. So I think that's exactly the point you were trying to make, that they're not the same, they're all different, there's a lot of variability, and clinically we have to learn about each one.
Yeah, no, I would agree with that entirely. And as I say, the thing for me, which it's really got me thinking really, is how much inhibition is necessary. And you know, whether it's 50% inhibition, 10% inhibition, 25% inhibition in terms of clinical effect. But I agree. We have to look at this in the whole in the clinic and make up our own minds.
And another question from Jens Gerwine has asked, are there JAK-specific differences in expression between different joints?
Gosh, that's a tough one. I don't know the answer to that. I mean, there's definitely differences between cells, as you might expect, and that to a certain extent explains why, you know, the inhibitors work differently, say in neutrophils and in CD4 T cells. It will just reflect the expression of the receptor. They're certainly upregulated in rheumatoid joints in general. We know that, and that's why they're a good target. But whether that varies between joints, I guess, yeah, inflamed versus uninflamed, they'll vary. But over and above that, I don't know. I don't think anybody knows, actually.
And lastly, do you think there's any mileage in developing increasingly specific, not 1 in 100, but 1 in 10 million specific, if you can? Do you know what I mean? Is there any mileage in that? They are combining TYK2s and JAK1s and things, but can you increase specificity further?
Yeah, it's a great question. I mean, it's very difficult to do. I wouldn't underplay the skill of these medicinal chemists. They use computer modeling. They use all sorts of things to play with the molecule. They'll know what an extra carbon looks like on an extra hydroxyl group, and so it's very, very challenging. I doubt it's possible to make something as selective as you're suggesting. But again, the other question I posed there is, which pathway do you want to block? It's interesting. The Japanese have gone for Paracetamy, which blocks JAK3, JAK1, which maybe wouldn't have been the most obvious pathway for many people, but it works. I've mentioned GM-CSF. Some people see that as a good pathway. Some see it as a bad pathway. Where we know, as you're probably aware, it's worth mentioning that TNF, for example, doesn't signal through the JAK-STAT pathway, so we can't block TNF this way, not directly anyway. IL-6 we're all familiar with. I'm sure we would all say IL-6 is a good cytokine to block because we block it in other ways. I quite like interferon alpha, actually. I think interferon alpha's got quite an important role in rheumatoid, which is not recognized as much as perhaps it might. But going back to the question, I think before you make that drug, you've got to be pretty damn confident that the pathway you want to block is the right one. And it may be we're thinking about this the wrong way. It might be the side effects and the adverse events that's more the differential between the drugs, because I'm sure you're going to talk about this now, Peter, but is there really a difference in the clinical efficacy between them? I'm not so sure.
That's a perfect segue. Thank you, John. If then put up my slides, we'll have a brief discussion, and save your questions, please, in the Q&A, and we'll tackle some more at the end of our discussion. So there's beautiful downtown Brisbane, and I'm just going to see if I can make these slides advance. Yes, I should be able to. We've already talked about our disclosures. I'm a humble clinician, and we've been doing lots of clinical trials with novel therapeutics for a long time. And really, I'm going to ask the question, JAK selectivity to be or not to be, that is the question. The rationale, as John's already said, are we limiting off-target effects? But there must be an advantage to selectivity to justify it. Is it efficacy? Is it safety? Is it convenience? Or is it cost? And we'll address some of those things. I had a look some time ago to see how popular JAKs were, at least in our country. And this is back when TOFA was launched in 2015, when Barry was launched 2018, and UPA really quite recently, April of 2020, and they took off. And now in our country, 1 in 3 patients are on a JAK, particularly the monotherapy market, and 1 in 3 patients who are NTXIR go straight to a JAK, and 1 in 3 who are TNF-IR go to a JAK as well. So I looked a little bit more recently. Here's when the oral surveillance press release was released. And really, as you can see, it hasn't affected TOFA, particularly maybe a little drop there. Barry's been very stable really over time. And UPA has taken off, pushing this efficacy from select compare, I suspect, but also the selectivity story. So we'll just see if that stacks up or not. So there's got to be an advantage clinically why these JAKs have been so popular. And I just put together a list. Oral versus parenteral is obvious. The cold chain can be a hassle, traveling, etc. These are small molecules, so they don't have immunogenicity issues. In many parts of the world, especially with a short half-life, can be stopped and started. Pregnancy, surgery, vaccination, COVID, infection, and their physiological effect and their pharmacological effect, although probably not the same in duration, is reasonably short. It does open the way for generics at some stage, and we'll mention that very briefly. Monotherapy efficacy is a real issue. In our patients, about 30% are intolerant or have a contraindication to methotrexate, and we've only got 1 IL-6 inhibitor in our market. And so being effective as a monotherapy agent is particularly helpful. They don't share the TNF kinds of things like inducing psoriasis, lupus, demyelination. Zoster is a clear signal, although the TNFs do get Zoster, so that's not 100% accurate. And John has already talked about metabolism and drug interactions and metabolites, etc. So let's think about that off-target effect. And John showed you this trying to separate efficacy from toxicity. So I thought I'd show you an example. And this comes from a Japanese study with low-dose background methotrexate. the Select Sunrise study where they had placebo and the low dose of UPA that we no longer can get and the high dose that we don't use. And they used ACR 50 as the efficacy, and you could see it plateaued around 15 milligrams a day. There didn't seem to be a huge benefit at the ACR 50 level of going to a higher dose. And they then looked at toxicity, looking at hemoglobin shift, which is a surrogate, if you like, for impairing JAK2 and erythropoietin pathway. And you can see as you push the dose, you did start to see a little bit more anemia instead of controlling inflammation and improving hemoglobin, suggesting that as you push the dose, exactly as John was saying, you might start to get some toxicity. So they picked 15 as the sweet spot between efficacy and those adverse effects, because as John has said, we would like to leave some of these growth factors alone and target different cytokines. And also, as he said, each one has its own cytokine fingerprint. This is looking at the 4 JAKs. It's looking at the different periods in hours of the day of the dosing interval, which cytokine, such as IL-6 or erythropoietin, which you want to leave alone, is inhibited for how long. And you can easily see that these are unique molecules. This is the more specific JAK1 being invented. This is being worked on in the Pfizer labs. This is the combo TYK2 JAK1. And you can see that individuals are different. We have to learn the fingerprint for each one, just like we learned that all the TNFs are different. And the numbers that John's already showed you, so I won't harbour the point, but they are clearly different, whether you use an enzyme assay, a cellular assay, whether you pick IC50 or IC20, they do have individual differences in the cytokine pathways they inhibit. And similarly, the time above IC50, as John has beautifully shown, changes. Now, I thought it was very interesting that while the stats look like they bind to different JAK dimers, stimulate signal transduction, I thought this was a lovely quote from a paper that said there's a certain degree of functional overlap between the stats. So the one stat which is supposed to transmit one dimer can be transduced by differences. So it's not as straightforward as the cartoons that we all teach show. Tremendous variability, even at the level of the stats. So that was just to reinforce what John's already said. Efficacy is the key for the clinician, of course, balanced by safety. Very comprehensive clinical trial program for all the JAKs, for all the clinical situations that you and I need information on. Where nobody's allowed to use treatment naive really around the planet, except in very rare instances. We all have to have methotrexate failures, and most of the regulatory authorities insist on that. But it does give us an inkling. If the world changes and cost is no longer an issue, and you have an oral generic agent that's as cheap as methotrexate, would you start with methotrexate if you had an alternative? That's a separate thing to consider. But clearly, MTX-IR is a common situation, as is BDMARD-IR. So rather than go through a million trials and try and convince you, I simply did something that you're not allowed to do, because this is an indirect comparison, different clinical trials, different background, comorbidities, disease activity, co-medications, etc. But I just looked at the CS-DMARD-IR patients. I took away the placebos, and these are the deltas for the 4 different drugs: ACR 20, 50, 70, low disease activity, and DAS remission minus less than 2.6. And I think really it's very hard to separate them on a level of efficacy when you're looking in this very common situation. There are small number differences, and that might be very clinically important, but it doesn't stand out as hugely different. This is looking at the BDMARD-IR patient population, and again, it's placebo corrected, and it's not allowed to do this comparison. Different trials, different background patient populations, disease activities, etc. But again, at the ACR 50 level, little bits at the ACR 70, low disease activity. It's hard really to separate them. You might argue that that is a big difference, and it might well be clinically significant, and that these numbers look good for this particular JAK in every situation in which it's employed. But really the numbers aren't hugely different, with a couple of caveats. And finally, the monotherapy patients, again, there are subtle differences, but they might be explained by the clinical trial population. I only did this because we're never going to get head-to-heads. These companies are not going to do head-to-heads, so we're going to rely on registries with real-world patients who could never get into a clinical trial because of comorbidities, malignancy, renal disease, cardiac disease, etc., past malignancy, a series of things. The registries are our best chance of seeing a surrogate for efficacy and not having adverse effects is keeping on drug for a long period of time. So efficacy, you could say, subtle differences. We'll never get a head-to-head. It's hard to see that they're very, very different. Now, safety is a huge issue, especially after oral surveillance, which we'll briefly touch on because it really is the elephant in the room. So we know that the JAKs have a series of things that we like to keep an eye on. All drugs, we need to make sure serious infection is not a major issue, different to what we expect with prednisone, methotrexate, and the other biologic agents, such as TNFs. We know there's a zoster signal because it interferes with interferon, as John has beautifully shown. But I was very interested that interferon gamma is not so much blocked by filgotinib. Interferon alpha was. And they, as we'll see, there seems to be less of a zoster signal given small numbers followed for a short period of time. We're trying to make sure all our girls now have HPV vaccination. And I've had a number of patients who complain of recurrent herpes simplex as a result of their JAK inhibitor. MACE, we've got to talk about malignancy. There doesn't seem to be a lymphoma signal. There does seem to be a non-melanoma skin cancer signal, not just with the JAKs, and all our patients have yearly skin checks living in the skin cancer capital of the world, but also methotrexate, TNFs, mycophenolate, azathioprine, cyclosporin. These are real issues across the immunosuppressive spectrum. VTE has been is a problem that has been clearly discussed and people worry about. Gastric perf, the numbers are tiny, and a number of lab abnormalities that we need to keep an eye on. These are all the issues that you and I have to look after every day with very little guidance from clinical trials, such as pregnancy, lactation, a reactivation of Hep B. There is now some data. So when you don't have head-to-heads, people do network meta-analyses using sophisticated statistical methods that I have absolutely no idea what they mean. The surface under the cumulative ranking curve, you rank clinical trials, you rank the number of patients, something like 1,500 patients in 7 clinical trials, and you come up with a ranking of ACR 20, and you come up with a ranking of serious adverse effects, and it's usually blown out of the water by the first head-to-head that you can ever do. And to be fair, the dermatologists are way ahead of us. Nearly all the new drugs are compared head-to-head against the gold standard. We've done a couple in psoriatic arthritis, we've done a couple in rheumatoid, but clearly head-to-heads. We might need to insist on them if we're going to move forward with novel therapies. So you can look at the big four: zoster, serious infection, malignancy, and MACE. And from the clinical trials, looking at event rate per 100 patient years, you can come up with these numbers. And the numbers have to be compared against what you would expect with methotrexate, low-dose prednisone, and no advanced therapy rheumatoid. Then you'll have to compare it against TNF inhibitors plus low-dose methotrexate plus low-dose prednisone. And really, the numbers don't stand out particularly different for MACE, malignancy, or serious infection, but clearly there's a zoster signal, as you would expect in an ACE inhibitor, but maybe not for this one particular JAK inhibitor, and we've seen that the interferon data already. So you'd expect in rheumatoid 3 per 100 patient years of serious infection if you're on methotrexate and prednisone alone, increases to 4 to 5 per 100 patient years if you're on a TNF, and these guys are in the same ballpark across the JAK spectrum. So I tried to pull out some data that's been presented at ACR last year. They're now out to nearly a decade of follow-up in clinical trials with tofacitinib. But again, we're talking 7,000 different patients. We're talking about 22,000 patient years of experience. There's the zoster signal that we mentioned. Everybody is aware of that signal. Malignancy didn't stand out, particularly different to what we expect from the British registry, from other registries, and quite different to oral surveillance, as we'll see. There's the lung cancer. There was no signal over this duration of time of those well patients going into this clinical trial program and staying in it for up to a decade. And there's the VTE signal, nothing that stands out. And there's the MACE signal, again, nothing much that stands out. But you have to be aware of oral surveillance. These were patients over 50 with a cardiovascular risk factor. It's been put a cloud over the whole JAK class, and thanks to the FDA, over every indication which a JAK is being used. And I just wanted to show you that none of this reached statistical significance. We'll just look at the TOFA 5BD, we don't use the 10, and out of 5,000 patient years for the two groups, TNF, adalimumab, or etanercept, depending which part of the world you lived in, there was either 47 cases or 37 cases, a difference that could be corrected if only 5 patients went from one side to the other. And if you looked at this patient population baseline demographics, there was a 5% difference in current smoking rate. There was a difference in cardiac aspirin rate. And if you just move 5 patients from there to there, that difference disappeared. Either way, you had to treat 587 patients to have 1 extra non-fatal myocardial infarct per year, and that caused a lot of hullabaloo. Even more with the malignancies. Now, I've told you there's a non-melanoma skin cancer malignancy signal for all the immunosuppressives, including TNFs, methotrexate, and the JAKs. And the difference here was, again, 62 events compared to 42 events. You had to treat 278 patients to get 1 extra malignancy in a year in this patient population. And people have gotten very, very alarmed at that. It's driven by the lung cancer difference. And I've already quoted you the smoking difference. So there was the difference. It was double the odds ratio, not statistically significant, but the incident rate was double. And the smoking rates in my country have plummeted since the government have taxed it out of people's pockets, and they can't afford to smoke as much. The rates have dropped significantly. But I live in the sun cancer capital of the world, and look at the difference in melanoma rates, TNFs, compared to this particular study population. There was many times more melanomas than there were in the TOFA group. I'd rather be cognizant of the melanoma rate. So all I'm trying to say is people have gotten very worked up over these numerical differences. I just wonder how clinically relevant it really is in a real world population. Perhaps the registries will be able to tell us, but there are the numbers. If we move on to baricitinib, and now some of these patients are out to 9 years of follow-up, we're talking about a couple of 3,000 people followed for something like 11,000 patient years of experience. Again, a zoster signal, as you'd expect. There really isn't much of a malignancy signal, 0.9. There isn't much of a MACE signal, 0.5 events per 100 patient years. And the big noise was over DVTs and PEs with baricitinib because of JAK2 inhibition. And again, this was driven by one of the early trials that had an increased rate. But over time, in this entire clinical trial program, there was no dose response, 2 milligrams to 4 milligrams. and the numbers are really quite reassuring over time. But again, this needs a mechanistic explanation. Is this a JAK2 inhibition effect, or is it a pan-JAK class effect? Is it a real effect, or is it a quirk of some of those trials? I think the answer is unclear, but either way, we don't give this stuff unless there's no other treatment available for patients with a history of VTE. But I have had to for people who failed everything. And even on occasion, I've had to anticoagulate the very, very rare patient to be able to give them a JAK safely. But that really needs a lot more understanding of the mechanism and a lot more understanding, again, from the registries. Is it real in real world patients with lots of comorbidities that would never get into a clinical trial? This is the UPA data. And again, they're fortunate enough to have comparators of adalimumab in the clinical trials, particularly select compare, but also some background methotrexate. There are some dose response effects at certain elements, but again, you could argue the numbers are not hugely different to what we would expect with rheumatoids on a biologic such as a TNF, what we'd expect with clearly a zoster signal, not much of a malignancy signal compared to adalimumab, compared to methotrexate. Again, similarly opportunist infections, very wide confidence intervals. So that's the kind of data that you can see. And I just threw in the psoriatic arthritis data. Again, a dose response 15 to 30, but really the numbers are consistent with what we've seen with all advanced therapies. They don't stand out particularly apart from zoster, no VTE signal, no malignancy signal. But again, we're talking 3 years of a relatively small number of patients in the psoriatic arthritis patient population. But that didn't stop the FDA having the rules that applied to one rheumatoid trial to answer for all different indications for the JAKs. Here's the Filgo data. This is out 4 years. Again, the numbers aren't huge, but again, 1.3 and 1.5 is not the numbers we're seeing with the other JAKs. I'd be interested in John's thoughts about the interferon inhibition and whether that's a true and a real difference. Again, this is looking at the PSA filgotinib data, and again, quite low zoster rates, small number of patients, only followed for 1 year, but that it might just be that filgotinib has less of those antiviral type issues. Now, the filgotinib has not been approved in my country because they didn't have the results of these 2 studies, which because in the lab, there's an effect on spermatogenesis with this drug. No one knows if it's reversible or not reversible. Is it real or not real from animal studies? But interestingly, in the press release from March of last year, and I haven't seen the clinical paper published, the placebo had a lower rate of 50% decline in sperm concentration than the treatment patients. And this led to this drug not being approved in my country. It's a shame. I can't wait to see the actual paper, and I can't wait to see the manta ray study fully published. There's a number of laboratory changes, lymphocyte count changes that you don't see across the board. You see them with one drug and maybe not the others. And lymphocytes, particularly NK cells, might be associated with serious opportunist infection risk. We try to keep the lymphocyte count above 500, like we do with leflunomide, azathioprine, cyclosporin across the board to reduce that risk. And similarly, neutropenia is what you'd expect with IL-6 inhibition. We don't see infection with it. It's a marginalization issue. Most of the drugs push the hemoglobin up. These other drugs, as you can see, there's minor differences, and this is upadacitinib on the end. It's an old slide. Some platelet changes. We do see some transaminitis, particularly if the patient's on combination of methotrexate. But again, I've not seen any issues of hepatic fibrosis or long-term effects. And similarly with the CPK, this is an oncostatin, JAK-STAT pathway issue, probably in the kidney. And again, I've not seen rhabdomyolysis although there's been the odd case report and a bit of myalgia. Basically, this is a biochemical change without having clinical efficacy, clinical impact. Some lipid changes like any IL-6 inhibition does. We're finding about a third of these patients wind up on a statin. We're very aggressive with statins because of premature atherosclerosis, but these patients respond to a statin and actually the atherogenic index doesn't change. And you'll find that controlling the inflammation is far more important in protecting atherosclerosis than any lipid changes, and there's some minor creatinine changes. So we've worked our way through, in a very short period of time, some efficacy data, some safety data, convenience, oral over parenteral, and they're all once a day now with the tofacitinib extended release, the UPA extended release once a day. And clearly, will there be a cost advantage? I think the biosimilars will rearrange everything. But right now, because this can be copied by clever chemists, such as in Bangladesh, you can get this medication made by Globe Pharmaceuticals for 50 cents a tablet. You can get Paracetamy for 90 cents a tablet. So the possibility exists when TOFA comes off patent in a year or two, that we can treat the poverty-ridden Asia-Pacific patient with cheap methotrexate, cheap JAK inhibition, and maybe we can actually make a big impact on rheumatic disease in countries where they can't afford advanced therapies up until today. So what's the EMA said? Over 65, smokers, other risk factors, only used if nothing else exists. I'm not sure the data really justifies that particular restriction, but clearly it's what everybody in Europe and England now has to work under, I imagine. And the FDA ruling, so all JAK class, all JAK indications were forced now to be post-TNF. And again, I don't know what they do with the monotherapy population. And I think that, again, that's a bit of an overreach, but maybe they considered safety is the most critical thing, while more of the long-term impact type studies are being done. So I think JAKs are here to stay. We just need to learn to use them wisely. And we need, like we always do, assess risk and benefit in the individual patient. We've got to be cognizant of risk factors and do something about them. Stop smoking. We've got to control lipids and blood pressure. We try not to give JAKs to those who have predisposing risk factors, such as history of VTE, and COX-2 inhibition was one of the other risk factors. But I think we have to be careful we don't throw the baby out with the bathwater. And at the same time, we shouldn't be a spin doctor making excuses and ignoring data and putting our patients at risk. So we need that balance. And I think that more data from registries will be very helpful. But as John has beautifully shown, these are complex systems with elaborate interlocking web of dependencies between components. We have to understand these relationships, and we have to understand these are unique drugs, and we have to learn how to use each individual one in particular. So I thank you for your attention, and we'll pass back to, please send your questions in to the Q&A box, and we'll hand back to Emma and John, and we'll ask for any questions, please. So we've got a couple of questions in the box there I can see. There's a question about the role of JAK inhibitors in other diseases than rheumatoid arthritis. We really haven't had a chance to go into that too much, but JIA, atopic dermatitis, alopecia areata, there's anecdotal stuff with dermatomyositis, interstitial lung disease. When I had a look on clinicaltrials.gov, there are 270 current clinical trials of JAK inhibition in unbelievable variety of diseases: Sjogren's, scleroderma, malignancy of every kind, myelofibrosis. So I don't know about you, John, but there's a huge array of things being experimented with.
Yeah, no, for sure, Peter. Yeah. Watch your space. I mean, let's hope the safety doesn't over, get on top of it all. I don't think it should. There's another question here, so let's do that, and then maybe we can have a chat about our own views on some of this. So there's a question about zoster vaccination. Are you using zoster vaccination before JAK inhibitors?
Yeah, what are you doing in your clinic, John?
We do check zoster IgG, so we check whether patients are immune to zoster. If I'm honest, I don't really understand the science of zoster. It doesn't quite make sense to me. So, but if a patient is zoster IgG negative, then we do consider vaccinating them. And of course, we really need a reliable killed vaccine to be completely happy with that. We don't like giving live vaccines to patients on immunosuppressive drugs. So we're increasingly screening people when they first get the disease and vaccinating them up front rather than leaving it till just before the JAK inhibitor. And
so we are a little bit lucky. We have Shingrix available in our country, even though it's not yet funded by the government. It's about $250 each injection by 2. We've had Zostavax provided free for anyone over 70. And I've been reading some of Kevin Winthrop's beautiful work, which showed that the risk for our rheumatoid patients are over 50, but our lupus patients are at a higher risk from their 20s, and we don't make a big fuss about vaccinating those patients. Perhaps we should. But so what we do, we try and get Shingrix into patients, as you say, well in advance of going on to advanced therapies, but we know it can be given at any time, even on therapy, if the patient's prepared to pay for it at the moment. And I think we'll push the government into providing it for all immunocompromised patients sooner rather than later. Zostavax is tricky, as you say. You've got to stop things for a couple of weeks. And Kevin and Jeffrey Curtis are doing a big study in not stopping and seeing how many patients run into trouble. So we'll have to wait for those results. But I think it is a real issue, and it's one of those things that we get our biologics nurse to tackle all vaccination as early on in the course of the patient's rheumatoid journey as we can. Flu, pneumovax, and HPV if appropriate, as well as zoster. So it's a conversation that we should be having earlier. So any other comments or queries? We're just about up for time. I would like to greatly thank you, John, for your time and for your beautiful presentation. I think that anyone who has an interest in JAKs, and will certainly be seeing a watch this space as far as JAK inhibition, selectivity, and what the regulators allow us to do. We're going to ask that this live stream version of this webinar will be available on YouTube channel, Cytokine Signaling Forum. Please fill in a short evaluation form to let us know your feedback. Thank you for your attendance. I hope you enjoyed the webinar. If you have any other outstanding questions, send them in and we'll do our very best to answer them and get them back to you via email or through Twitter. And you can get slides kits, you can get publications on the cytokinesignaling.com website. It's a very useful resource. The next edition of ForumLIVE will be looking at precision medicine and rheumatoid arthritis on Wednesday, the 15th of June. I recommend you tune in for that. And again, you can look at it on YouTube at your own leisure. And with EULAR around the corner, there's going to be lots of sessions at EULAR, and there's going to be the highlights from EULAR 2020 on the 7th of June. I thank you so much, John. It was great to catch up, and I'd say good evening or good morning to everybody, and take care.
Thanks, Peter. Thanks for hosting so well. Thank you. And thanks to all attendees.
All the best.